Hi, so far we've been talking about the acquisition and reconstruction of fMRI data. We talked a little bit about experimental design last time, and now we're going to move into pre-processing. So we're going to talk about pre-processing for a couple of modules before moving on to statistical analysis. So prior to statistical analysis, fMRI data undergoes a series of pre-processing steps, which are really aimed to identifying and removing certain artifacts and also validating certain model assumptions. So in general, the goals of pre-processing are: 1, to minimize the influence of data acquisition and physiological artifacts; 2, to check statistical assumptions and transform the data to meet these assumptions; and 3, to standardize the location of brain regions across subjects to achieve validity and sensitivity in group analysis. So if we're going to perform a group analysis, we need our different brains to align with one another. So here's an illustration of the pre-processing pipeline. And note that when we perform an fMRI experiment, we collect both structural T1 images and functional T2 star images. And so the pre-processing is performed both on the fMRI data and the structural scans. Throughout this lecture and the next one, we will talk about all these different steps, and so we'll revisit this slide towards the end. So what are the pre-processing steps that we want to perform? Well, we want to perform visualization and artifact removal, something called slice time correction, motion correction, correction for physiological effects, so co-registration, so we want to align the functional and structural images to one another, normalization, so we want to align all the images to a stereotactic space, spatial and temporal filtering. So let's start talking about visualization and artifact removal. And this is really the first part of the pre-processing pipeline. And really this is something that you should do after every step of the pipeline. We should always visualize what happens after you do a certain pre-processing step to ensure quality control. And so what we do is we use exploratory techniques to investigate the raw image data and detect possible problems and artifacts. And so one artifact that's very common in fMRI data is that it often contains transient spike artifacts as well as slow drift over time. So here we often use an exploratory technique such as principal components analysis, or PCA, and use that to look for spike-related artifacts. Here's an example showing transient spike artifacts in the data that were acquired at certain volumes across time. And these artifacts are due to that the gradient performance was affected during the acquisition of some of these EPI images, these echoplanar images. Okay, and so what we see here is that in certain slices of the brain are affected, and so you see that they're a bit wider than the other ones. And so these artifacts, we want to be able to know where they are as well as be able to remove their effects. The next thing we would like to do is we often do something called slice time correction. And so in this case, what we're doing is recall that when we collect these three-dimensional volumes of the brain, we often do it one slice at a time, or almost always we do it one slice at a time. Basically, what happens is that each slice is then sampled at a slightly different time point vis-a-vis each other. So the first slice that you collect is at time t, and then with a certain lag, the next slice is, and then next slice, and then next slice. But typically when we analyze the brain volume, we think of it as being collected all at the same time, but that's not really true. So slice time correction, what it does is it shifts each voxel's time series so they appear to have been sampled simultaneously. So let's take a look at the effects of slice time correction. Suppose you have 3 slices and you have a single voxel in each of these 3 slices. And let's just assume, for sake of argument, that each of these voxels has the exact same signal in it, this maroon signal here we see in the figure. Let's suppose that slice 1 is collected first, thereafter slice 2 is collected, finally slice 3 is collected. What will happen is the voxel in slice 1 will be measured at the green time points along the function here. The voxel in the second slice will be acquired during these yellow time points, and finally the voxel in slice 3 will be acquired during these blue time points. So if you don't do slice time correction and just treat them as being simultaneously acquired, what happens is that you're basically going to get an estimated HRF, or the shape of an HRF that looks quite different between these 3 slices, even though the underlying signal was the same in all 3. And so what you can do is you often need to correct this using some sort of temporal interpolation. So the idea behind temporal interpolation would be that you use information from nearby time points to estimate the amplitude of the MR signal at the onset of the TR, for example. And then you use some sort of spline or linear or sinc function in order to shift it so that they're measured at the beginning of the TR or the middle of the TR or whatever you choose as your target. You can also use ideas from Fourier analysis to slide the time course by applying what's called a phase shift. So slice time correction, a lot of people don't perform slice time correction. Instead, incorporate the differences in measurement time in their subsequent models. And so we'll talk about both of these ideas later on when we talk about statistical models. One thing that you have to correct for is head motion. So even very small movements of the head during an experiment can be a major source of error if not treated correctly. Because remember, we often look at a single voxel of the brain and we extract the time course, the intensity at that voxel at each time point to get our time series. Now, if a person is moving back and forth during the scans, between scans, then when analyzing a single voxel, that voxel is going to shift back and forth. So across time, we might be looking at different voxels as we progress across time because of movement. This is a very serious problem because then we don't really know what we're analyzing, but head motion can usually be corrected using what's called a rigid body transformation. So in head motion correction or motion correction, the goal is to find the best possible alignment between the image that you're looking at and some target images. And so what you do is you try to align the target image and your input image with one another. And so to do that, you have to transform the input image so that it overlays the target image, more or less perfectly. And so how do you do that? Well, in motion correction, we typically use what's called a rigid body transformation. And so a rigid body transformation has 6 variable parameters. You can shift the input image in the X, Y, and Z directions, and you can also rotate it in 3 directions, and these are often called pitch, roll, and yaw. So you can rotate them around the 3 axes as well. Now a rigid body transformation assumes that the shape and the size of the brain is static across the different time points. The only thing that changes is its position. And so while that might not technically be true, it's probably a good enough approximation, and it seems to work pretty well in performing motion correction. So rigid body transformation isn't the only type of transformation that we could perform. In general, there's a lot of different things that we can do to the images. So one, we've talked about translation. That means a shifting of the image in space. And so in this cartoon, we're shifting it in a 2-dimensional space, and we can do it along the X and the Y axis here. We could also do rotations, and so here there's one rotation that we can do. We can do scaling, that we keep the same shape, but we just make it bigger. And we can do something called shearing, where we kind of pull it down in different directions. And that's a way of kind of stretching the image in different ways. Each of these different types of operation can be performed in different transformations that we perform on our brain. There's different types of linear transformations that are often used. The one we just discussed for head motion is a rigid body transformation that has 6 degrees of freedom, so 6 variable parameters. So we can translate in 3 directions, and we can rotate around 3 axes to match our input and target brain. A similarity transformation is the same. We do translations and rotation, but we also have a single global scaling. So we can increase or shrink the size of the brain, but keep the general shape. Finally, we have affine transformation, which has 12 degrees of freedom that allows for 3 translations, 3 rotations, scaling in 3 dimensions, and shearing in 3 dimensions as well. Finally, we don't have to use linear transformations. We can also use something called warping, and these are transformations where the equations related to the coordinates of the images are all nonlinear, and they're much more complicated, and we'll kind of brush over them for now. So in motion correction, the target image is typically defined to be the first image of the fMRI time series, or alternatively the average over all of them. And so the goal is then to find the set of parameters which minimizes some cost function that assess similarity between the image and the target. And so examples of these types of cost functions could be the sum of squared differences or the mutual information. So what we want to do is we want to make the target image and the input image match as close as possible. Okay, let's assume that this green oval is the target image. And let's suppose that we have another image, which is this purple oval. The goal here of motion correction is now to align them with one another. So basically we want to translate and rotate this purple image until it aligns perfectly onto the green image as follows. Once we do that, we can look at the translations and rotations that we needed to make at each time point and study that over time. And then we get these types of plots that show kind of the history of movement across time for each image. So here there's 300 different images, and the two plots here show on the top we see the X, Y, and Z shifts, and in the bottom we see the rotations in 3 dimensions. And so basically what you can see is that the movement across time, and particularly at time around 160, there's a shift in the Z direction. Next, let's turn our attention to the structural MRI image that we collected in the beginning of the session. So the next thing that we do is we typically register this to the fMRI images in a process referred to as co-registration. So we want the structural image and the fMRI image to be as aligned as possible. Now, why do we want to do that? Well, for one reason, it allows us to visualize single subject task activations on the individual's anatomical information. So while we analyze the fMRI data, we want to discuss the results looking at the structural images, because the structural images allows us to make out where exactly the boxes we're studying are located, while the functional contains the data that we want to analyze, but it's hard to make out boundaries between different anatomical regions. Another reason we do co-registration is it simplifies later transformation of the MRI images to a standard coordinate system. We'll talk about that next time. So there's certain key differences between co-registration and motion correction. In both cases, there's similarities as well, because in both cases we want to align 2 images with each other. But the difference is that the functional and the structural images don't have the same signal intensity in the same area. So one is maybe a T1 weighted image and the other a T2 star, and so the intensities are going to be quite different from one another. So you can't simply subtract them from one another. So you can't do a least squares type of fit to find the best fit between the two images. Also, their shapes may vary, so we can't do a rigid body transformation because the shapes of the images might be somewhat different. So what we need to do in co-registration is to use at least an affine transformation, so use 12 degrees of freedom that allows us to not only move it around in space, but also scale and shear a little bit. And we also have to do mutual information cost functions because we have the differences in signal intensity. Here's an illustration of some normalized data taken from, and this output is from the popular SBM software package for analyzing fMRI data. And so these are on the right we see the functional data, and on the left we see the structural data, and this is after they've been normalized. And so now you can see that they're basically mapped onto each other. Okay, so that's the end of this module, which is the first of 2 pre-processing modules. Here we'll discuss things like visualizing your data, performing slice time correction, motion correction, and co-registration. In the next module, we'll talk about normalization and also a little bit about spatial smoothing. Okay, I'll see you then.